Hi, I'm Sarah, and I'm a LURA student in the Stem Cell Engineering Lab, and I'm going to present my summer project. Leukemia is a type of blood cancer that starts in the blood stem cells existing in the bone marrow. Did you know that almost every patient carries the risk of return of disease or relapse after treatment with intensive rounds of chemotherapy? This is because most cancer drugs are designed to kill rapidly dividing cells. However, not all leukemic cells are created equal. In addition to the large pool of differentiated leukemic cells, there is a small fraction of quiescent leukemic stem cells. These cells do not divide rapidly, so they can survive chemotherapy effectively and regrow the disease. Leukemic stem cells have similar properties to the hematopoietic stem cells that are involved in the production of normal blood system. They both exist in the bone marrow, but they reside in different microenvironments. Local microenvironment of stem cells or stem cell niches are very important in regulating different biological functions of cells, such as their survival, proliferation, differentiation, and homing capabilities. However, the process of how these stem cells interact with their niche is not fully understood. Investigating molecular and cellular determinants of this interaction can help us find unique features of leukemic stem cells that can be targeted in the clinical setting. On the other hand, integrins are cell adhesion receptors on the surface of the cells. They mediate the interactions between cells and their extracellular matrix. Structurally, they are heterodimeric complexes containing an alpha and a beta subunit. They can interact with unique ligands in the extracellular matrix and transfer signals to start signaling cascades to regulate multiple biological functions. These adhesion molecules retain stem cells in the niche, so it is important to further investigate the role of integrins in the population of stem cells to explore their influence in drug sensitivity and adhesion-mediated chemoresistance. This project compares the gene expression profile of integrins between populations of leukemic stem cells and normal hematopoietic stem cells using bioinformatics methods. To do so, a microarray dataset was selected that analyzed genome-wide expression of 4 control groups of hematopoietic stem cells and 9 groups of leukemic stem cells derived from patients with acute myeloid leukemia. Then, GO2R analysis tool was used to analyze the dataset. This tool allowed for identifying genes that are differentially expressed across groups of samples. Results from this tool was presented as a table of 3,000 differentially expressed genes ordered by their significance, and their small program was coded to screen for the genes related to integrin subunits. It was found that several integrin subunits were significantly downregulated in the leukemic stem cell populations. In conclusion, this bioinformatic-oriented data analysis can provide a direction for future investigation of integrin protein levels and their relevance in the stem cells to potentially provide a better understanding of leukemogenic mechanisms and ultimately designing therapeutics that specifically target leukemic stem cells in order to reduce occurrence of relapse and improve how we treat cancer.